cuticle is divided into two types. One is non-chitinous epicuticle, means without chitin cuticle, and another one is with cuticle, that is called chitinous procuticle. See, it has an epicuticle. Epicuticle is nothing but epicuticular layer of insect is about 1 to 140 141 in thickness, and in general, which covers the entire external surface of an insect. Epi means above. Cuticle is the endoderm, epidermis, like our skin in human beings. The same way in the insect, we have a epicuticle and endocuticle. This epicuticle is the 1 to 141 thickness of the insect, which covers the external surface of the insect, except chemoreceptor cells, which will not cover the chemoreceptor cells. Except this epicuticle covers the entire surface of the body of an insect, including midgut ends of gland cells. The main function of epicuticle is to prevent water loss from the insect body. The cuticle is to prevent the water loss from the body of an insect. So that is the major function of the epicuticle.
Sir, your voice is not audible to us.
Is it audible? Hello?
Yes, sir, now audible.
Okay. So the cuticle, function of cuticle is to prevent the water loss from the body of an insect, and it also maintain the water content in the body of an insect. It could be terrestrial or it could be aquatic insects. So this epicuticle again divided into 4 layers. That is called, one is outermost cement layer. Outermost cement layer. See, the epicuticle again divided into 4 layers. Outer one is called outermost cement layer, and second one is called wax layer. The cuticle layers. Cuticle again consists of 4 layers. The first layer is outermost cement layer, and second layer is wax layer. Then third layer is called outer epicuticle, and fourth layer is called inner epicuticle. These 4 layers which are present in the epicuticle region. For example, if you take the outermost layer, that is called a cement layer. The cement layer consists of carbohydrates. This is the outermost layer of the epicuticle. This cement layer is consist of carbohydrates, proteins, and long chain lipids, and which consists of some polyphenolic substances. So the cement layer, distribution of cement layer may vary within an insect. So cement layer is different in different kind of insect. So it may be totally absent. The cement layers may be present in some insect. It may not be present in some insect. For example, if you take honeybees, in honeybees cement layer is not present. So like that, this layer is varies with depend upon the insect. The function of cement layer are to protect the wax layer from operations and to act as a reservoir of mobile lipids. The function of this cement layer is to protect or to reserve the lipids in the body of the cuticle of insect. So this is the function of a cement layer. This cement layer is the outermost layer of epicuticle of the insect. Then next we have a second layer. That second layer we can call it as wax layer. Below the cement layer, you will have another layer, the second layer, which is called wax layer. Wax layer usually lies below the cement layer and provides a kind of waterproofing. Means this wax layer acts as a waterproofing to protect the cuticle from other desiccations. That's why it acts as a waterproofing. In this, the wax canals are considered the route of entry into the insect for oil soluble compounds. The composition of waxes varies not only between individual of the same species, but also within the same individual of the different time of development. So these waxes can vary from species to species in the insect. So the waxes are mostly composed of, this wax layer is mostly composed of hydrocarbons of 12 to 31 carbon chain lengths. This wax layer is composed of hydrocarbons. And also this composed of some esters and some kind of fatty acids and alcohols. So that is about the wax layer. Wax layer is the second layer of the epicuticle. Then you have an outer epicuticle. The outer epicuticle is the third layer of the epicuticle. The outer epicuticle is a trilaminar layer. So 12 to 18 nanometer in thickness and present universally, except to over sensory endings. So except to sensory endings, all over this outer epicuticle is present. Lines the trachea, it also present in trachea, serves as the insertion point of muscle tonofibrillae. So that is called outer epicuticle. Outer epicuticle is a trilaminar layer, a thickness of 12 to 18 nm, so which is present universally except the sensory endings. The next we have a inner epicuticle. This is a fourth layer of epicuticle. The inner epicuticular layer consists of a polymerized lipoproteins. This is composed of a polymerized lipoproteins and stabilized by quinons. It is produced by secretion of epidermis. This inner epicuticle is produced by secretion of epidermis. Probably serves as a reservoir of the extracellular enzymes associated with repairing of the wounds. So this inner cuticle is provide the enzymatic activity for repairing mechanism. So that is about the three layers of the epicuticle of the insect. Then you have a chitin. How chitin is synthesized? So what is the chitin? Already I told chitin is a polysaccharide. So let us see how chitin will be synthesized in the insect cuticle. Chitin consists 25 to 60% of dry weight of the insect cuticle. So chitin is a very stable polysaccharide. Chitin is a kind of polysaccharide, largely made up of N-acetyl D-glucosamine. So this chitin is a hard substance which are made up of polysaccharide, which is present in the epicuticle of the insect. These are made up of N-acetyl D-glucosamine and some glucosamine. These are linked up by a 1 to 4 A-glucosidic linkages. These are approximately 1000 monomers. Polymers or polysaccharide is nothing but a linkage chains of many monomers, that is called 1000 monomers per chain. And every sixth monomer may be glucosamine instead of acetyl glucosamine. Its structure may be considered as a substituted cellulose. If you observe the polysaccharide structure, it's similar to the cellulose structure, with N-acetyl moiety replaced by the OH on carbon 2 found in the cellulose. So this is the structure of the chitin. So then we have a chitin synthesis. How this chitin layer, how this chitin is synthesized in the insect cuticle. Synthesis of chitin for the cuticle takes place in the epidermal cells. Generally, the synthesis of chitin takes place in the epidermal cells of the insect. Like a trehalose are nothing but a glucose is converted into uridine diphosphate. First, this trehalose or glucose is converted into uridine diphosphate. Chitin synthesis in insect appears to be similar to that occurring in a fungi. So in the same way, fungi consist of a chitin. Like in a similar way, this chitin also synthesized in the insects also. The pathways and biosynthesis pathways are same that of fungi. Lipids in a cuticle. Lipids they do not contribute the rigidity of the cuticle to protect the insect against dehydration. So lipids also present in the cuticle. This will help to dehydration and are important to survival of the insect. The major portion of the lipid is found in the wax layer of the epicuticle. So mostly the more lipid is found in the waxy layer of the epicuticle. Already I told waxy layer is the second layer of the epicuticle. In this waxy layer, more lipid is formed. This layer is the principal barrier to the passage of water into or out of the insectic cuticle. So this layer acts as a barrier to proceed water inside and outside of the insect cuticle. Then you have a hardening of the cuticle. The hardening of the cuticle starts with the stabilization of protein in cuticle. There is a stabilization proteins which are present in the cuticle, which makes the hardening of the cuticle by the formation of cross links between the molecules. So these are the, we have a terminal amino group, peptide chains with a free amino group or dibasic amino acids. Usually lysine is amino kind of amino acid as a result of tanning, and cuticle becomes the formation of synthesis of hardening of the cuticle. So there is a pathway of chitin synthesis. How exactly the chitin synthesis takes place in the. Let us see the diagram, the pathway of chitin synthesis. For example, trehalose. Trehalose in a chemolymph first this trehalose gets converted into glucose by an action of an enzyme called trehalase. The action of an enzyme trehalase, this trehalose gets converted into glucose. By the presence of exokinase and enzyme, this glucose gets converted into glucose 6-phosphate by the action of an enzyme or the presence of an enzyme exokinase. Glucose gets converted into glucose 6-phosphate. Then again the glucose.
Where we get trehalose,
Hello?
sir? Trehalose.
Trehalase is one kind of glucose in the insect cuticle. So this glucose phosphate isomerase gets converted into fructose 6-phosphate. So glucose 6-phosphate gets converted into fructose 6-phosphate in the presence of glucose phosphate isomerase. Then again fructose 6-phosphate gets transferred into glutamine fructose 6-phosphate and aminotransferase. By the presence of this enzyme, it gets converted to the fructose 6-phosphate into glucosamine 6-phosphate. Again, this glucosamine 6-phosphate, this forms the N-acetyl glucosamine 6-phosphate in the presence of glucosamine 6-phosphate N-acetyl transferase enzyme. Then again, this N-acetyl glucosamine 6-phosphates gets converted into N-acetyl glucosamine 1-phosphate by a presence of an enzyme phosphoacetyl glucosamine mutase. Then again, this N-acetyl glucosamine 1-phosphates gets converted into uridine diphosphate N-acetyl glucosamine by presence of an enzyme iridinophospho N-acetyl glucosamine phosphorylase. So likewise, this uridinodiphosphate N-acetyl glucosamine forms the chitin synthetase. By the presence of chitin synthesis, chitin will be formed. So that is called N-acetyl glucosamine. So this is a process or a pathway of synthesis of chitin in the insectic cuticle. So this is the pathway of chitin synthesis. It is very important for chitin synthesis in the examination point of view. Then again you have a degradation of chitin. That is a synthesis of chitin. Then next you have a degradation of chitin. What is meant by degradation? Degradation of chitin is, tell me. Degradation of chitin is carried out by the chitinolytic system, which consists of two enzymes. One is chitinase poly beta 1,4, 2-acetamido and 2-deoxy and D-glucoside glucoramide hydroxylase, which hydrolyze the chitin polymer. Degradation is nothing but to catalyze the activity or breakdown activity of the chitin, which is called degradation of chitin. So the polymer of N, distyl chitinobasis, which is subsequently hydrolyzed by chitinobasis beta 1-N-acetoglucosamine or chitobasis acetamidoxylate hydroxylase to the chitinolytic system has been shown the various insect. What happened? The chitin is degraded by the presence of an enzyme chitinase and to form a beta N-acetyl glucosamidase. This is called the enzymatic. Tell me.
So please explain in the board if possible. On the board. Blackboard.
Board, we cannot cover all the topics on the board, I think. Because I have to cover every chapter for even one period. So in the board, only 1 or 2 subtopics will be covered. So that's why I'm trying to cover
understand the synthesis and
yeah. The synthesis, that is the only pathway which the stepwise cascade reactions present in the. If you go through the textbook, you will understand it. The diagrammatical representation present in the textbook. The trehalose, first from the trehalose, it's get converted to chitin. There is a cascade biosynthetic mechanisms involved in the synthesis of chitin. Once you go through that, you will understand it. Otherwise, you cannot cover the topic, all topics. If I go only with the synthesis of chitin on the blackboard, it only takes 40 minutes to 1 hour. That's why I'm trying to cover all the subtopics. Okay. If you have a doubt, you just read out, you'll understand it. The next, penetration and distribution of insecticides. Penetration and distribution of insecticides. What is meant by penetration? Anybody have an idea?
Absorption into the cuticle.
Any toxicant has to enter the body.
Entering of an toxicant. Entering of an toxicant inside of an insect body, which is called penetration. Distribution means supplying of the toxicant throughout the body is called distribution of any chemical substances supplied throughout the body of an insect, which is called a distribution. So that is called penetration and distribution of insecticides. Any toxicant has to enter the body of the living organism and react at its site of action in order to perform its activity. Whenever the toxicant enters into the body of an insect or any organism, it goes to its target site or target organ and it performs its activity. The first barrier of the contact insecticide is the insectic cuticle. If any chemical substance or any insecticide has to enter inside of an insect, it has to cross the cuticle of the insect. That is called the insectic cuticle. While the stomach poison has to pass to the stomach poison also pass through the stomach, it has to cross the gut wall. Like there are some kind of barriers. Then after entering the body, an insecticide has to cross the several barriers before it reaches the primary site of action. During the process, a part of it may be reversibly absorbed by all kind of tissue components. Some of the chemical substances or some of the insecticides will detoxify and some will be excreted and either unchanged or after the metabolic conversion. Some insecticides need to be activated before they can react with the target. See, some insecticides can perform their activity inside of the insect body. If it is not detoxified, it performs its activity. If it is detoxified, it cannot perform its activity. And if some of the chemical substances or some of the insecticide will be excreted through the body, so it cannot perform its activity because there will be no insecticide inside of the target organ. So like that is called that kind of penetration and distribution of the insecticide, and this distribution also depends upon the metabolic activity of a particular toxic or body of the insect. Then we have a cuticular penetration and ingestion. So cuticular penetration will have one theory. It is the most important theory, that is called lateral transport theory. Lateral transport theory. This lateral transport theory is proposed by Gerlach in 1969. The lateral transport theory, according to this theory, insecticide use the trachea. According to this theory, many insecticides use the trachea of an insect as a port of entry and reach the target by diffusing through the outer layer. The trachea have a direct access to central nervous system, that is the CNS. Mostly, I already told in the offline classes, mostly all the insecticides CNS, and because they are lined with the cuticle tissue, a continuous layer between the outside of an insect and target site is available for insecticide to diffuse the target without being forced to cross one or more membrane barriers. So there is a proving of two scientists. The two group of scientists are in favor of this theory. The group of scientists does not agree that Chemolymph act as a carrier insect body. So Chemolymph is present in the insect, which helps in the circulation of the particles or circulation of the circulation takes place in the insect body, which can carry the insecticide to the target organ. So one group scientist does not agree. Chemolymph cannot carry the this insecticide into the body of an insect. The other gives direct evidence of lateral movement and entry via trachea. The first group argues that if Chemolymph delivers the insecticide at the target organ, the permeability of cuticle must be high enough for the toxicant to reach the Chemolymph in quantity of sufficiently large to explain the observed toxic effect. The extremely small rate of penetration of both, for example, dihydrin, it is a kind of insecticide, a polar and methamyl polar through isolated cuticle does not fulfill this condition. Secondly, any manipulation of the concentration of insecticide in the hemolymph of the following given topical dose should influence the toxic effect elicited by this dose. See, example, if olive oil is injected into the hemocoel of an insect, for example, if you take the olive oil is injected into the hemocoel of an insect, it should absorb the lipophilic insecticides from the hemolymph and lower the KT50, that is called knockdown 50, such an effect has not been demonstrated. So mostly this olive oil is a lipid kind of oil which is mostly absorbed in the lipophilic insecticides. But whereas water and other, it cannot absorb to the lipophilic. Lipophilic means kind of fatty lovable substance, lipid lovable substances. But thirdly, if the toxicant molecules reach the target via hemolymph, administration of equidic toxic doses either by topical application or by injection should result in the same hemolymph concentration. So it was found that hemolymph concentration of methamyl following injection in houseflies was much larger than that of the following topical administration. See, topical administration is nothing but the toxicant should given the outer surface of the body by a cuticle. But whereas the methyl concentration should be given directly through the injection into the body of houseflies. So that concentration of the injected one is much larger than the topical administration. So during the whole observation period, finally, if hemolymph is the carrier and if it is circulation is prevented, it should not allow the transport of toxicant to the target site, and the insecticidal compound should be harmless. So what happens if hemolymph is a carrier? If you stop its circulation, what happens if you stop the circulation? The insecticides or any pesticides cannot transport the toxicant substances to the target site. So it cannot affect the body of an insect. So it acts as a harmless toxicant. So that is a theory which was proposed by Gerlach. That theory we can call it as a lateral transport theory. Then we have again another theory you have that is called hemolymph transport theory. Hemolymph transport theory. The other view is more like that one that is a general accepted in studies of drug action in mammals. According to this, the toxic compound is absorbed by diffusing through and not laterally in the integument, and after crossing this barrier, it is conveyed by hemolymph to the target organs, to several other organs where it is accumulated and metabolized or excreted. This concept also applies to toxicants that are ingested and thus arrive the target after passing through the gut wall. So any toxicant when go to the chemolymph, then it could be some toxicant could be accumulated and some toxicant could be metabolized and some toxicant could be excreted out when it is reached through the gut wall and to reach the target site. So in support of this theory, the evident advanced is that hemolymph of cockroaches, that is called Periplanet americana, treated topically with a LD dose, that is called lethal dose of diazoxin. Diazoxin is a one kind of insecticide. This LD dose of perithrin one contained sufficient insecticide to explain the observed disturbances of the neurophysiological functioning of the CNS. The internal distribution of these compounds is mediated by hemolymph transport. So many scientists believe that both routes may operate depending upon the property of the compound in a solution, the nature of the insect. Most insect toxicologists agree that the cuticle areas around setae and cincinae articulate the membrane, offer less resistance to the diffusion of insecticides. The cuticular structure also agrees it's upon. In addition to this, toxicant can enter through the dermal gland ducts and oral canals. So hemolymph transport theory explains that the toxicant can cross through the hemolymph of the insect. Then some of the toxicant can penetrate through the gut wall. That is called penetration through gut wall. Insecticides do not have insect body by penetration only. This insecticides not only enter the cuticle, it also they may also be ingested with food and reach the hemolymph. Any insecticide can enter. Some insecticide may enter with the cuticle, and some insecticide may enter with the food substances, and they will reach the hemolymph. And from there, it reaches to the target and non-target organs after passing to the gut wall. So that is called penetration through the gut wall. Then you have a mechanism of penetration and distribution. So penetration of the toxicant into insect body involves partitioning of the
Not audible, sir.
Hello. Hello, is it audible?
Yes, sir. Now
Now
audible.
it is.
Is it okay?
Yes, sir.
Yeah, mechanism of penetration and distribution. Penetration of the toxicant can into insect body involves partitioning of the chemical between the lipophilic and aqueous phases. Since many contact insecticides are lipophilic, many insecticides are lipophilic. They love the lipids because this cuticle is made up of lipid membrane. So if the insecticide is a lipophilic, they easily cross the cuticle of an insecticide. So that's why these many insecticides are lipophilic. It is surprising that polar insecticide appear to penetrate the cockroach Periplaneta americana integument more rapidly than lesser polar ones. For example, this means the compound should be sufficiently lipophilic to dissolve in the outer lipid layer of the cuticle. If it is a insecticide is a lipophilic, definitely it dissolves in the layer of the cuticle sufficiently. Hydrophilic to move into the more polar inner layers of the cuticle. Hydrophilic cannot cross, but lipophilic can cross sufficiently. So the partition process which determine the rate of penetration of an insecticide are likely to be affected by method of application. For example, phosphoric acid applied to cockroach cuticle in water. If you apply the phosphoric acid to the cockroach cuticle in water, it did not penetrate. It did not penetrate until the water has to evaporate, because the phosphoric acid is not a cuticle is not a water philic, it is a lipophilic. So the phosphoric acid cannot penetrate the cockroach cuticle when if it is dissolved in water with phosphoric acid. So this water has to evaporate, then only the phosphoric acid will enter the cuticle of the insect. Other hand, when chemical is applied in organic solvent, the solvent may lead to removal of the surface wax, the point of contact of the spray. This can affect the rate of transport. There are different kind of mechanism of penetration and diffusion. For example, some factors determining the cuticular penetration rates. Cuticular penetration rate. For example, effect of solvent. some dissolvents, different kind of solvents, some chemical substances dissolve in different solvents. For example, the effect of solvent. The solvent that is used for administering an insecticide plays a significant role. In T. splice Glossina astinae, the rate of penetration and the rate of internal accumulation of organochlorine and organophosphorus compounds vary for various solvents. For example, DDT accumulated fastest when applied in diacetyl ketone. If DDT is applied in diacetyl ketone solvent, it accumulated very fast. And slowest, the same DDT shows the action is very slowest when applied in methyl volate solvent. See, the rate of penetration is depend upon the effect of this solvent. Example, DDT accumulated very fast when applied in diacetyl ketone solvent. The same DDT shows the slowest action when applied in methyl volate. So the insecticide accumulation depends upon the effect of the solvent. The insect used in this study do not to metabolize DDT and difference in the rate of internal accumulation, therefore directly reflect the differences in the rate of penetration. For example, acetone, solvent widely used in laboratory studies. This acetone is widely used in laboratory studies, appears to be a rather ineffective vehicle. Acetone and non-dudicane, when tested in application solvents for digoxin on houseflies, showed that N-dudicone, high boiling hydrocarbon, digoxin had a higher initial penetration rate in comparison to the acetone. The difference between the solvents was found to be strain dependent with respect to acetone. Dudicane increases the initial penetration rate to 5-fold in a stain with the so-called pen factor, that is inheritable factor for the reduced penetration rate, but less than twice in a stain without this factor. The same pair of solvents were tested for their effect on the speed of knockdown of houseflies by perithrin and bioaristamine. So it was found that perithrides also penetrate faster. When compared to the perithrid, perithrid is one kind of insecticide. Already I told this perithrid penetration is very faster with dedicon than with the acetone. If you apply the perithrid in a solvent of dedicon, this penetration rate of perithides will be very faster than compared with the acetone. If you apply the perithrid in acetone, the penetration rate will be very slow. The same perithrid will apply in N-dudicane solvent, the penetration rate will be very high. So the studies tells us that the insecticide penetration rate depends upon the effect of the solvent. That is called factors determining cuticular penetration rate. Then you have a cuticular composition, the penetration and distribution in relation to selectivity. So penetration in relation to resistance. Then we have a effect of physical agents on cuticle. So what are the physical agents that affect on the cuticle of an insect? So the waterproof cuticle prevent desiccation of the insect body. Waterproof cuticle which prevent the desiccation of the insect body. The waterproofing of the cuticle is affected by physical agents. Physical agents nothing but one is abrasion and second one is surruption. Abrasion is nothing but the epicuticular lipid layer may be lost. See, epicuticle is made up of a lipid. If that lipid layer will be lost through simple physical actions such as a apersion. Apersion is nothing but rapid movement జరగడం వల్ల క్యూటికల్ లాస్ అయిపోయి దాంట్లో ఉన్న డెబ్రీస్ కానీ లేదంటే దాంట్లో ఉన్నటువంటి డస్ట్ పార్టికల్స్ అన్ని కూడా ద ఎగ్జోస్కెల్టన్ పైన డిపాజిషన్ కావడాన్ని అబ్రేషన్ అంటాం. The water barrier of the cuticular lipid must be absorbed out. Allow the water beneath to escape. So the insecticidal efficacy of dust against some adult grain infesting beetles traveling among dusted wheat grains increases with increasing hardness of abrasiveness of the particles. So the insecticidal effectiveness of inert dust is correlated with the hardness of the material. For the inert dust to be effective, they must have a hardness number of 6 or above on Mohs scale, particle size below 1 nm with particle ranging between 1 to 2 nm of being most effective. The abrasion is nothing but if lipid layer is lost above the epicuticular layer, the hardness substances, dust particles, all will enter the exoskeleton and they will be deposited, and then it act as an insecticide to the particular insect. So that is called the abrasion. Then we have a sorption. This is one kind of a physical agent which act on the cuticle of the insect. Then next we have a sorption. Sorption is the idea that abrasiveness means losing of the lipid layer of the cuticle and deposition of the hardness and dust particles on the insect. The idea that abrasiveness was the mechanism by which inert dust kill the insect prevailed for many years. Whatever the dust, whatever the hardness which are deposited by losing of the lipid layer of the epicuticular region of the insect will harmful to the insect. Sometimes which causes death of the insect. Sorption is nothing but absorption or adsorption or ion exchange of the deposited dust particle, which is called sorption. That absorption of the deposited dust particle and the hardness of the cuticle, which causes to kill the insect, and it makes lethal to the insect, which is called sorption. Evenly, it was indicated that sorption property of inert dust is more important than abrasiveness. Sorptive dust were indicated to be effective against insect as early as 1940, but because of the prevailing belief that abrasion removed the epicuticular lipids. Abrasion is nothing but removal of the epicuticular lipids, and the lack of knowledge of the physicochemistry of the lipids. This fact was not widely recognized. Certain charcoals and clays with poor hardness, therefore, have low insecticidal activity. So, deposition of some charcoals and clays also takes place in the epicuticular regions, but they cannot make the insect to kill. It has a lower insecticidal activity. Is related to other sorptiveness. So this is all about the sorption. Sorption is nothing but absorption and adsorption and exchanging of the ions of a dust particle. This makes the insect to kill, which is called sorption and absorption. These two are the physical agents on the cuticle of the insect that makes to kill the insect. There is a effect of kaolinite and 3 mineral derivatives obtained by progressive acid and heat activation against the adult tribolium confusum. Adult tribolium confusum is the one kind of insect, a kind of beetle kind of insect. So this physical characters will change according to the heat activation. For example, these are the derivatives from minerals. Kaolinite is a derivative of mineral. H-halosite is a derivative of mineral. Meta-H-halosite is a derivative of mineral. And mullite is the derivative of mineral. So then activation of temperature means heat activation. Then there would be some physical characteristics, lower absorption and lower sorption, lower absorption and high sorption. So depending upon the activation of the heat. So that is all about the effect of physical agents on cuticle. The factors affecting action of physical agents. For example, other factors also affect the efficacy of inert dust, like electrostatic charge and atmospheric humidity. For example, between two silica aerosols, dry dye 67 is generally slightly more active. See, dry dye 67 silica aerosol generally slightly more active than dry dye 68. The primary difference between two substances is the presence of 4.7% by weight of ammonium fluosilicate in dry dye 67. This addition gives a particle as a positive electrostatic charge, which enhance their ability to deposit and adhere to the insect and increase their effectiveness. See, silica aerosol dry dye 67 is generally more active than dry dye 68 because dry dye 67 consists of or presence of in a substance of 4.57 weight of ammonium silicate. So if 4.257 of ammonium silicate present in dry dye, it shows a high effectiveness of the insects. So that is this is also one kind of a physical agent which affect on the action. So then next we have a effect of chemical agents on cuticle. So those are the physical agents which affect on the cuticle. Then in the same way you will have a chemical agents, how these chemical agents act on the cuticle. Many chemical agents adversely affect the cuticle and consequently the survival of the insects. Chemical agents nothing but petroleum oils and solvents are the simplest form of among them. They probably exert their effects by causing water loss to the disruption of the cuticular wax layer. They could either dissolve in the wax or upset the orientation of the wax molecules, which are normally highly organized in their case. So there are many insecticidal methods, for example, conventional insecticides and insect growth regulators. Conventional insecticides. The insecticides including organophosphates, chlorinated hydrocarbons, carbamates, pyrethroids, and nicotinides and protinides are primarily these organophosphates, chlorinated hydrocarbons, carbamates, pyrethroids, nicotinoids mostly affect on the nervous system of the insect. These insecticides, you can call it as a nerve poisons, with the exception of protinides. Most of them are act on the nerve poison. They disturb the oxidative phosphorylation in the insecticidal action of the compounds is very rapid. It is highly unlikely that inhibition of chitin synthesis. These insecticides like organophosphate, chlorinated hydrocarbons, and carbamates and pyrethroids, we can call it as a neurotoxicant. This plays a significant role in killing of the insecticide on the nervous system of the insect. Then next we have a insect growth regulators. Insect growth regulators. The insect growth regulators, which include juvenile hormones, that is called JHS. These are the insect growth regulators, one of the hormones like juvenile hormones. And these compounds that mimic the action of growth hormone, that is called benzophenylurase, and other compounds interfere with the normal formation of the
Sir.
cuticle.
Excuse me, sir. Hello, sir. Hello.
Hello?
Can you explain juvenile hormone? Can you explain it?
Juvenile hormone is the one kind of hormone which is present in the insect, which help us to formation of the cuticle in the insect, like human being hormones, like a growth hormone. This is the same way the juvenile hormone is present in the insect, which helps to formation of the cuticle. Then we have a chitin synthesis inhibitors. Chitin synthesis inhibitors.
Okay.
There we have seen the formation and synthesis of chitin. Chitin synthesis takes place by trehalose, and that gets converted to glucose and glucose 6-phosphate. Then finally we got the chitin formation. So now here the chitin synthesis inhibitors. Chitin synthesis inhibitors is nothing but to stop the synthetic activity of chitin, which is called chitin synthesis inhibitors. For example, during the course of the development of new herbicides in early in the 1970s, scientists at Philips Dover in the Netherlands combined two herbicides, diclorvinyl and diurin, in an attempt to create a super herbicide. The resulting compound 1,2,26-dichlorobenzyl and 3,4-dichlorophenyl urea was totally inactive to herbicide, but was toxic to insect. More interestingly, it appeared to act in an entirely different manner than insecticides that were in use that time. In the mortality was invariably connected to the process of molting, and the adulting stage was not affected. Further exploration led to development of an 1,4-phenyl and 2,6-difluorobenzyl urea, commonly known as now a difluorobenzoin. These benzoin ureas interfere with the deposition of normal cuticle, apparently by affecting the chitin synthesis. The effect of these compounds appeared to be inhibition of biosynthesis of chitin in larvae of Pteris brassicae. Pteris brassicae is a kind of insect. So this inhibits the biosynthesis activity of chitin, means to stop the activity of biosynthesis in the chitin larva of Pteris brassicae. So the protein content in the cuticle is not affected by the difluorobenzoin treatment, whereas the chitin content are markedly decreased. So chitin content is markedly decreased, gradually decreased by the stopping of the biosynthesis of the chitin. The insect of chitin synthesis are not inhibited by difluorobenzoin and other benzophenyl urea. Difluorobenzoin has no effect on chitin synthesis. It is inactive as a chitin synthase inhibitor. Hence, the chitin synthase inhibitors do not act on the polymerization step directly. For example, another group of compounds represent yet another mechanism of interference with normal clitorization process of the cuticle. The inhibitors of aromatic L-amino acid and decarboxylase of dopa decarboxylase, a key enzyme in cuticle scleralization, are able to kill the dipteran larva at the mouth. These inhibitors, example, alpha-methyl dopa causes the abnormality in the lipid-rich outer epicuticular layer. In larva treated through feeding, a newly formed cuticle is apparently defective as a water permeability barrier, since it permits the free movement of water in both directions, as shown by the water loss and water uptake experiment. The treated larva probably die as a result of dehydration. So, a naturally occurring chitin synthesis inhibitors in the root of a tropical medicinal Plumbago has been isolated and characterized as different as a plumbagin. This compound inhibits the This compound inhibits the equidiasis of 4 lepidopteran pest species. Equidiasis of the 4 lepidopteran pest species. So this is one act as an inhibitor. So Pectinophora grassifera and Elanthus versina and Elanthus. Concentration of 0.0. It causes 29% of reduction in incorporation of glycan to form a UDP glycan into precipitate that was collected on glass filters, which was presumed to be chitin. See, here the chitin synthesis inhibitors are there. For example, chitin synthesis inhibitors are plumbingen, this plumbingen, which inhibit the equidiasis of 4 lepidopteran pest species. So equidiasis means shedding of the old skin of the insect, which is called equidiasis, which inhibits this equidiasis process. So these are called the chitin synthesis inhibitors. For example, diflubenzuron and benzophenylurase. These two are the chitin synthesis inhibitor to stop the synthesis of chitin. So that is all about the insecticide cuticle. So insecticide cuticle, mostly probably questions they may ask in the exam. See the model examination questions. What do you understand by the lateral transport theory? Proposed to explain the penetration of insecticide insect the body. So you just go through the lateral transport theory, so you will understand. So they may ask in a short kind of question that is called lateral transport theory. Then we have a how do the physical agents affect the insecticide cuticle? The abortion and corruption, how does physical agents will affect the insect cuticle? So that is also one question they may ask in the exam. Then we have a next question that how do the synthesis and degradation of chitin takes place in the insect? So the process of synthesis, how the process of chitin synthesis from trehalose to the chitin. There are a cascade reactions which are involved in the formation of the chitin. Once you go through the synthesis of chitin and also go through the how the chitin will be degraded from chitin synthesis. So go through the synthesis and degradation of the chitin in the insect. The next question you will have a what are the various factors affecting the penetration of the cuticle? So there are many types of factors that will affect the penetration of cuticle, for example, solvent and other things. So you once you go through the various factors, how that affects the penetration of the cuticle. That may ask in the long question. Next question you have, how does the hardening of insect cuticle takes place after equidiasis? So they may ask in a short type of question. So these are the questions they are going to ask regarding to the insecticide cuticle. Okay. Once you go through these questions, you will understand it. It's a little bit, the toxicology is a little bit a confusion like a subject. It is new to you people. Once you go through, you will understand. If you have any doubt regarding to this, you may ask in the next session. Once you read it.
Okay, sir.